So chapter 19, genetic technology. So this is a very interesting chapter because this is a chapter where you learn that when you learn the basic knowledge in biology. Now in this case here is the structure of DNA and also the process of gene expression from DNA to become mRNA, mRNA to become the protein. So from this knowledge itself, how a scientist put in their creativity, okay, to play with the DNA and to play with this process to produce products that are needed by human, and also to create processes or procedure that are useful for human. So either the product is useful or the process is useful. It's all designed based on the basic knowledge. So this chapter is an application of biology for you to see when you have the basic knowledge, how do you apply to help the human society? It's very interesting. So the learning outcome you need to learn here is define the term recombinant DNA. So I'll read the learning outcome first. I'll explain in today's lesson. Explain what is genetic engineering. Explain the 3 methods to obtain the gene to transfer into organism. And explain the roles of these enzymes that are used in genetic engineering, GE. So since this chapter's name is called genetic technology, so at least we need to know what is the meaning of genetic technology lah. So genetic technology, the word technology means technique. Technique means methods. So these are the methods that are used in genetic engineering, creation of genomic libraries of DNA, and the DNA fingerprinting. Now, out of these 3, right, in this chapter, you only learn this method, which is genetic engineering. So genetic technology involves these 3 techniques, but we only learn the first one, which is genetic engineering. So I will not explain the other 2, which are not in the syllabus. Okay, so we'll continue to explain what's genetic engineering from here onwards. So genetic engineering, so when the word engineering comes to your mind, right, when the engineering, this word comes to your mind, what idea do you have about engineering? What idea do you have about engineering? Do you build something? You're trying to take the bricks, take the cement, okay, take the steel or whatever and try to build something out of it, and you design it in a way that whatever you build, it depends on the design, right? How you put things together. So genetic engineering have to do with you working with the genes, right? So working with the genes, try to do something, rearrange them and so on. So the definition of genetic engineering is an artificial way. Now, every time you come across artificial means it's man-made. So it is not natural. Okay, so it's an artificial way of changing the genetic information of living organism. So how do we change the information? By transferring a gene from an organism, which is called the donor. So we transfer the piece of gene from this person, the donor, to another person, the recipient, or can be another organism like bacteria or plant and so on. So these are the recipient. So it can be of the same species or different species. So from human to human, or from human to bacteria, human to plant, human to whatever. So this is called genetic engineering. Okay, so that the recipient organism will then express the gene. That means the gene is expressed to become the polypeptide that's coded by red. Polypeptide. So say, for example, if you transfer the human insulin gene to the bacteria, so the bacteria will now be called the transformed bacteria. Transform because you change it already. What do you change? You change the genetic makeup. So that's why it's called the transformed bacteria. So this bacteria will now start to express insulin gene and produce insulin. Or if you transfer human growth hormone to this bacteria, it will start to express this gene and produce human growth hormone. Human growth hormone gene, please add the word gene there. Or you may want to transfer blood clotting factor 8 gene, okay, to the lamb. Then the lamb will then start to express this gene and produce blood clotting factor 8. Now, this lamb that receives the foreign gene is called the transgenic organism because you already transfer the foreign gene to this organism. So it's a transgenic organism, or the more familiar name that you know is genetically modified organism, GMO. We have already modified the genetic information of the organism by adding this foreign gene from the donor to the recipient. So the recipient organism is called genetically modified organism. Okay, so take note, for bacteria, we call them as transformed bacteria, but for other organisms, we call them as transgenic organism or GMO. Okay, now recombinant DNA. So what is recombinant DNA? If you look at the word recombine, so it is actually a piece of DNA made up by artificially joined 2 or more DNA pieces together. Each piece comes from a different source. So it can be same species or different species. Okay, so that means you got a piece of DNA, and this piece of DNA actually comes from 2 pieces from 2 different sources. So this can be from person A, this can be from person B of the same species, or this can be from human, and this one can be from the bacteria. So the resultant DNA, after you combine the 2 DNA from 2 different sources, is called the recombinant DNA. So now, in order to do genetic engineering, we know that we're supposed to work with the genes, right? So every process that you want to do with the gene, you have to use enzymes. So you know that when you want to work with the molecules in the body of the organism, we need enzyme to catalyze any processes to change that, to change that DNA. So therefore, we will straight go and look at what are the toolkits that are needed for us in genetic engineering so that we can carry out all the processes to obtain the gene from the donor and transfer the gene to the recipient. So what are the toolkits for genetic engineering? Of course, the very first one is enzymes, right? Because enzymes can catalyze a lot of processes to change the DNA or to change the gene. So these are the 4 enzymes that you need to learn in the syllabus: restriction enzymes, DNA ligase, reverse transcriptase, and DNA polymerase. So we're going to discuss this one later on. Then number 2, we need vectors. What is vectors? Vector is a vehicle. Vehicle means it's like a car, right? Because you want to transfer the gene from one organism to another organism, you need a transporter. Okay, you need the transporter to transport the gene from donor to recipient. Okay, so therefore, vector is actually the vehicle that can artificially transport the foreign genetic material into the recipient cell. So transport from here to here. So that's called the vectors. So the vector in genetic engineering can be plasmid, can be virus, can be liposome. Now, you have also come across the word vector when you learn about infectious disease, because this word vector in general, it means the vehicle. It means the vehicle. So when you say what is the vector, for malaria, then we know it's actually the female Anopheles mosquito, right? It's the organism that can carry the pathogen from an infected person to an uninfected person, right? So it's a carrier. So it is a vehicle. So that's the general name for the word vehicle, general meaning for the word vehicle. So in GE, vehicle is actually a vector is actually the vehicle to transport this gene to the recipient cell. And number 3 is the marker. Marker means marker is actually a gene that we need in order to find out which cells have been transformed, or which cell in the organism has received the gene, the foreign gene. So marker It's like when you write your notes, right? You use marker to actually highlight those words, right, that are important. So marker here is for us to highlight among all the bacteria that I have, which bacteria cell has taken up that foreign gene. So that is the marker, to mark the bacteria that has taken up the foreign gene, or to mark the recipient cell that's taken up the gene. So that's the marker. So today we will focus on enzyme and vector only. We'll learn marker in some other lesson. So firstly, let's look at the enzymes needed for GE. First one is restriction enzyme or restriction endonuclease. Now this enzyme comes from bacteria. And what do this enzyme do in the bacteria is, now just like human, we can be infected by bacteria and viruses, right? And bacteria can also be infected by virus, because virus is smaller than bacteria. So usually what does the virus do when they infect a cell? Is like the virus, okay, will actually inject its genetic material into the whole cell and then convert the whole cell into a viral production factory. So that's why we said now the bacteria is being infected by the virus, which is the host cell. So some bacteria, they will resist the infection by virus. Even though the virus can inject its genetic material into the bacteria has got enzyme that will cut this DNA of the virus. So therefore, it will no longer be able to convert this bacteria to a viral production factory, because the DNA is being cut already. So what do they use to cut? They use this enzyme to cut. So that's why this enzyme is called restriction enzyme, because this enzyme restricts the infection of the bacteria by this virus. And this virus that infect the bacteria is called the bacteriophage, or phages in short. Bacteriophage or phages in short. Hang on. So how do we name those restriction enzyme? Before that, now restriction enzyme is also called restriction endonuclease. Endonuclease. So nuclease is an enzyme that catalyzes the hydrolysis, okay, of the nucleic acid, means the DNA. So it will actually cut the DNA. Endo means from inside. Now, usually you know that when you got polymer molecule, because DNA is also polymer of nucleotides. So these are all nucleotides, right? So usually when we want to actually hydrolyze a polymer, we will actually start to remove the monomer one by one from the terminal end, isn't it? So we remove one by one. But restriction enzyme, they do not cut by removing from the ends of the chain. They actually cut right in the center of the chain. So therefore, it's called endonuclease, means it will hydrolyze inside. Endo means inside the entire chain. It does not remove the nucleotide one by one from the terminal nucleotide. So this is the terminal nucleotide. So that's why it's called endonuclease, cutting the DNA from the inside. So the enzyme, this enzyme is given name based on the bacterial name and a Roman number. So the bacterial name is given in short form, and the Roman number indicates the number of enzyme that it has been discovered in this bacteria. So if this is the first enzyme, it's called number 1. Second enzyme discovered is called number 2, and so on. So for example, EcoR1 is a restriction enzyme that comes from the bacteria E. coli strain RY1. So that's why EcoR1. 1 stands for the first enzyme discovered in this bacteria. So what does this enzyme do? In order to cut the DNA, it must first bind to the DNA. So the enzyme must bind to the substrate, right? So it must bind to the part of DNA that it will want to cut. So the part of DNA that it will bind to is called the target site of this enzyme. So the target site of the enzyme, if you look at the next slide, so these are the example of the enzyme, and these are the target site of this enzyme is very specific because they will bind to that target site based on the base sequences there. So each enzyme have their own target site with specific base sequence. And the base sequence, as you can see from here, is between 4 to 6 base pairs. And it's very interestingly, if you read this base sequence, right, you look at the first example. If you read this chain of DNA from the left to the right, GGATTC, the base sequence will read the same in the complementary strand from the right to the left is also GGATTC. Can you see? The rest of them are the same, GAATTC, GAATTC, AAGCTT, AAGCTT. You can see all of them, right? When you read the base sequences of the target site in one strand of the DNA from left to right is same as the complementary strand from right to left. So this property is found in the target site. This property is called palindromic. So all target sites are palindromic. Next, when they cut the DNA, so when they cut DNA, they can cut the DNA in two ways. You can see here. This is first way, and this is second way. So you can see in the first way, they cut in a staggered manner. So staggered means it's like a staircase like that. And the second way, they cut straight down. It's not staggered. So when they cut in a staggered manner, you see that the DNA that's produced will look like this already. So that means this part here is double-stranded. This part here is double-stranded. This part here is single-stranded, or I can write like this is even easier to understand. Okay, so when you cut, your DNA will look like this, whereby there's this region of DNA. There's this region of DNA here, and this region of DNA here. This is not clear. Let me do it again. Hey, where's my DNA? Gone. My DNA gone already. So when I cut, okay, like this, right? So do you see that this region here is single-stranded, and this region here is single-stranded? So these two regions that are being produced after cutting, they are called the sticky ends. So the sticky ends are the short single-stranded DNA found at the end of the double-stranded DNA, and it's called sticky ends because the bases here are unpaired. So they are able to stick to another complementary strand. So that's why they're called sticky ends. So when the enzyme cut in a staggered manner, it will produce sticky ends on the DNA. Now, if the enzyme cut in this manner, there'll be no sticky ends. So what do we call those ends here if it's not sticky? So these are the DNA that's being produced. There's no sticky ends, right? So these ends are called the blunt ends. So these ends here, okay, they are called the blunt ends. Blunt ends because there's no bases that are unpaired. So it's blunt ends. So very interesting. This is a property of the restriction enzyme. So coming back to here where we stopped just now. So let us revise again what is target site. Target site is the site where the restriction enzyme will bind. It contains specific sequence of bases, and it's always 4 to 6 base pairs. And mostly the base sequences will be palindromic. That means the sequence of bases from left to right in one strand will be equal to sequence of bases from right to left in the complementary strand. Example here, GAA, TTC, GAA, TTC. So within the target site, this is the target site, right? Within the target site, the enzyme will cut at the restriction site. So let's say, for example, this is a restriction site. So the restriction site is exactly where the enzyme restrict means will cut. Target site is where it will bind. So there's slight difference between the meaning of target site and restriction site. Okay, so when it cut, it will produce either the sticky ends or the blunt ends. So all the words are the definition I've mentioned just now. Okay, sticky ends and blunt ends. So second enzyme you look at is DNA ligase. Now you already learned DNA ligase is an enzyme that we use during DNA replication, right? To join the Okazaki fragments together, remember? So DNA ligase is an enzyme that's used to join DNA fragments together. So in this case here, for example, you got two DNA, one on the left and one on the right. And both of them got sticky ends. And these two sticky ends are complementary because the A can pair with the A, the T can pair with the A. Sorry, the A can pair with the T can pair with the A. So they're complementary sticky ends. So at low temperature allow hydrogen bond to form. So when you put these two DNA together in the same test tube at low temperature, then the sticky ends will bind together by complementary base pairing. And they'll form hydrogen bonds. So then you can see all these hydrogen bonds are formed already. But the sugar phosphate backbone here, can you see the sugar phosphate backbone? They are not joined yet. So therefore, there's a gap between the two sugar phosphate backbone of the two DNA fragment. So this gap is called the nick. So therefore, we need an enzyme to catalyze the formation of phosphodiester bond between this sugar phosphate backbone to join these two DNA together. So DNA ligase is being used, okay, so that the nicks at the sugar phosphate backbones are being sealed by forming phosphodiester bonds. So in genetic engineering, where exactly will we use DNA ligase is this example here. So we obtain insulin gene from the human, and then we obtain a plasmid from the bacteria. So we isolate the insulin gene from human cell and the plasmid from bacterial cell. So we got two DNA from two different sources. And then we cut both of them with the same restriction enzyme. Same restriction enzyme. So when we cut with the same restriction enzyme, we will create complementary sticky ends. So you must cut with the same, then only you will produce complementary sticky ends. So this sticky end can actually join to this sticky end. This sticky end can join to this sticky end, right? So when you put the insulin gene and the plasmid together in the same test tube, then you see that this sticky end will form complementary base pair with this sticky end, and then we'll use DNA ligase to seal the nick by forming phosphodiester bond, and as a result, we'll get the recombinant DNA. Right? So DNA ligase joins DNA fragments by sealing the nicks at the sugar phosphate backbone and to form phosphodiester bond. So DNA, which is the gene here, will be added to DNA, which is the plasmid, to form rDNA, which is a recombinant DNA. So this is another diagram to show the same process where we cut the plasmid with equal R1, and then we cut the gene with equal R1, and then we put the plasmid and the gene together, and therefore we allow the sticky ends to join. The resultant product can be the recombinant DNA, but some gene did not join to the plasmid, some plasmid did not join to the gene. So therefore the product will be of 3 types: the original plasmid, the recombinant DNA, and also the gene. Now this part here is not in syllabus. If you're interested, you can read. That means when you cut and produce when you cut and produce blunt ends, how do you make sticky ends to it? How do you add sticky ends to it? Because without the sticky ends, the 2 DNA molecules cannot bind. So if you cut and produce blunt end, you have to add your own sticky ends. But this is not in the syllabus, so I'll skip this whole thing, okay? Now, the last 2 enzymes that you will learn is actually the reverse transcriptase and DNA polymerase. Now, this one is very easy. You already know one. Now, reverse transcriptase catalyzes the process called reverse transcription, right? So if transcription is DNA to mRNA, reverse transcription should be reverse in direction, right? That means it is converting mRNA to DNA. So that's called reverse transcription. So if we use reverse transcriptase, that means we are trying to use mRNA as a template to synthesize the single-stranded complementary DNA. So let's look at the diagram. So we have mRNA as the template. And then from this template, we want to synthesize a complementary strand from this mRNA, but we want to synthesize DNA. So this DNA is a single-stranded DNA, right? And it's complementary to the mRNA. So what do we need? We need this, later on you'll see. So after you synthesize a single-stranded cDNA, we have to remove this mRNA. So we hydrolyze the mRNA so that we only left with the cDNA as the template. And then using the cDNA as a template, we will then synthesize the complementary strand so that eventually we get a double-stranded DNA, or in short, dsDNA. So this is a process of synthesizing double-stranded DNA from mRNA. So what do we need? Step number 1, in order to convert mRNA to cDNA, we need a primer, reverse transcriptase, and nucleotides. So you see, in mRNA, right, we need to add a primer, means it's a short single-stranded DNA or RNA molecule. So in this case, we use DNA. So we use a DNA primer to form the 5' end of the new DNA chain. So after that, only the reverse transcriptase enzyme is able to add 3 DNA nucleotides 1 by 1 to the template, to the mRNA template by complementary base pairing. Because reverse transcriptase is similar to DNA polymerase, it cannot join nucleotide to nucleotide. It must join nucleotide to an existing DNA. So that's why you use a primer first to be added to the template. In this case here, the RNA template. In this case here, DNA primer. Then only reverse transcriptase can add the nucleotide, DNTPs, deoxynucleoside triphosphate. So after step number 1, the single-stranded cDNA is being produced already. We will add RNA nases to hydrolyze the RNA. So we remove the RNA. So we left with only the cDNA as a template. So using this template, we will then synthesize its complementary strand. So we will need a DNA polymerase. We need nucleotides, okay, to synthesize it. Right? So we have looked at the 4 enzymes that are needed in genetic engineering. where the restriction enzyme will cut the DNA. DNA ligase will join DNA fragments. Reverse transcriptase and DNA polymerase are used to synthesize double-stranded DNA from mRNA as a template. Now, the question is, now, in order to understand this question, there are 3 ways to obtain a gene in order to transfer into the recipient organism. So based on the gene expression, right, from DNA, transcription into mRNA, translation into the polypeptide. So based on this gene expression, there are actually 3 ways we can obtain the gene. Of course, the very first way you can think of is we go to the cell, we go into the nucleus, and we try to fish out that gene, for example, insulin gene. We try to fish out the insulin gene. That means from the DNA, you try to get the DNA in the nucleus of the cell. Okay, this is the first way. Second way is, I don't want to get the gene from the nucleus of the cell. I say that since this cell is going to express insulin gene, it's going to produce many copies of the mRNA. So inside the cell, there will be many copies of this mRNA for insulin, but in the nucleus, there are only 2 genes for the insulin gene, because one in paternal chromosome, one in maternal chromosome, right? So between the 2 genes that I want to fish out versus the many mRNA in the cytoplasm, which one is easier for me to isolate? The mRNA, isn't it? Because you've got more copies, it's easier to isolate. So I can try to isolate mRNA from the cell, and then from mRNA, I synthesize the DNA. So this is second way of producing, obtaining the gene. From mRNA, you produce the DNA. So this is the first way. This is second way. Now, third way, of course, is starting from the polypeptide chain. Now, because this cell produces insulin, there will be a lot of insulin inside the cell. And the insulin can also be secreted out of the cell. So it's actually quite easy to isolate the insulin. So when we get the insulin, we can use the insulin to produce the DNA. Now, how do we do that? From insulin, we find out what is the amino acid sequence in the insulin. From amino acid sequence, we will work out what is the codon, the triplet codon sequence in the mRNA. And we work out what is the base sequence in the DNA. And after that, we key in this information into a machine. And this machine is a DNA synthesizing machine. So we key in this information, we put the nucleotides into the machine, and then the enzymes into the machine. The machine will then synthesize this DNA for us. So therefore, there are 3 ways we can obtain the gene. From DNA to DNA, from DNA to, from mRNA to DNA, from the protein to DNA, 3 ways. Now, among these 3 ways, right, the best way is from mRNA to DNA. Can you see that the best way is from mRNA to DNA? Because a lot of mRNA that we can easily isolate from the cell, and then from mRNA, we can then convert into DNA using enzymes. So it's very straightforward. Whereas from the insulin, there are many steps involved, because first thing, you need to find out the amino acid sequence first. And after that, you need to find out what is the codon sequence in mRNA. And after that, you need to find out what is the base sequence in DNA. Then only you can put in the information to the DNA synthesizing machine to synthesize DNA. So many steps involved. So this is slower. So the best way is the second way. Okay, other than that, there's another reason. So I'm going to explain to you here. So if the question asks you, what is the advantage of making the gene, which is the double-stranded cDNA, starting from mRNA, as compared to isolating the gene from the genome, starting from protein to synthesize the DNA? Now, have I explained the word genome? You can write into your notes. Genome is actually all the genes and genetic material, all the genes and genetic material in a cell or in an organism. Okay, so all the genes or genetic material in a cell. In a cell or organism. So in a cell, where can you find the genetic material? In a cell, where can you find a genetic material? Lipei?
Nucleus.
Nucleus, very good. Other than nucleus, where else? Januli?
Mitochondria.
Very good, mitochondria. If in plant cell, where else? Losisian? Yes. Plant cell? Plant cell? Losisian? Chloroplast, very good. Chloroplast. So when you come across the word called genome, genome means all the genes and genetic material in a cell organism. So it will be the DNA in the nucleus, including mitochondria as well as the chloroplast. Okay, so that's the meaning of the word genome. So let's look at answering this question. So what is the advantage of obtaining the wanted gene starting from mRNA is, number one, because the cell that expresses the gene contains large quantity of mRNA, so it's very easy to isolate. And then it's actually harder to isolate the gene from the genome because inside the genome, let's say in the nucleus, there are 20 to 25,000 of genes. So if you go to a nucleus and you try to fish out 2 insulin genes, it's actually very difficult to find one. It's that you go into a hall. This hall got 25,000 people, and you try to find 2 people without using your handphone run and computer. It's very difficult to find. Okay. And also one more thing. You see, in the process of gene expression, when you start from DNA, right, the gene in the DNA contain both exon and intron. So after transcription, we will form primary RNA transcript, and this primary RNA transcript contain both the intron and exon. So the next step is actually processing. In the process of processing, we will have to remove the intron and join the exons together. So this process is actually called RNA splicing, cutting and joining of the RNA. Because intron is the non-coding region of gene, it doesn't code for the polypeptide chain. So we have to remove them in an additional step called RNA splicing. That is excision of introns and joining of exons. So if we start from fishing out the gene in the DNA, that gene will contain intron. And what happens is that bacteria cannot remove the intron because bacteria has no enzyme to carry out RNA splicing. So when we insert this piece of gene to bacteria, we will not get insulin because the bacteria will be reading the entire DNA, producing the mRNA, and then reading the whole thing to form a polypeptide, which is actually not the insulin already. So we cannot just fish out the gene and paste it into the bacteria because bacteria cannot carry out RNA splicing. So we get something else. Okay, interesting, right? Okay, so we have looked at the enzymes as the toolkit in genetic engineering. So now we will look at